Thanks for attending Rogan's PhD dissertation presentation. So this is a summary of all of his work. So Rogan joined us a couple of years ago from Nebraska Lincoln. Before that, K-State for his bachelor's degree. And pretty soon here, he'll be moving off back to the plains, the prairies, the North Dakota State, right? Yeah, Rogan's had a great contribution to our lab. Research, extension, even some instruction. So really checking out all those like land grab mission areas. So really appreciated yeah, Rogan's contribution. Of course, we wish he stayed a little bit longer, but we do wish him well moving forward. Yeah, award-winning, especially from the stakeholders, like really got a lot of recognition from stakeholders during his brief time here. FAR fellow, Project Apis M fellow, Southern IPM Center Student Award. We need to get a photo of this. He knew he had this award, but here's his lovely plaque there. It's been collecting dust on my desk for like 6 months now, but they did require a photo, so we'll get to that as well. So yeah, I think we'll just leave it to Rogan. He'll have, I guess, 40-45 minutes, maybe a presentation, and then there'll be a chance for some Q&A and comments afterwards. So take it away.
Yeah, so first I want to thank everybody for coming. I'm Rogan Tokach. This is my dissertation defense seminar, and it's titled Developing Sustainable Parasitic Mite Management Strategies for Beekeepers. And these are my two main culprits that I'll be talking about today, and I'll be giving a lot of information on them. But just to start out with a little bit of a background on honeybees, I don't think it's a surprise to anybody that honeybees are very important pollinators. They're responsible for billions annually through their crop pollination services. Of course, a lot of that pollination services coming in areas like Central California with the almond pollination, but also in areas like Washington with apples, in that northern Great Plains region with North Dakota, and then down that Mississippi River Delta. So honeybees are very important pollinators. But we also probably, that honeybees are facing very high annual colony losses. Now, I want to point out this doesn't mean that the overall number of honeybee colonies is decreasing in the US, but we're seeing that beekeepers are consistently reporting losses of really over 40% since 2018. Now, beekeepers are having to recoup these losses by doing things like splitting their colonies to really get back up to their original numbers. And eventually, as these losses continue to climb, and I believe last year it was even over 50%, this is becoming unsustainable. And these beekeepers are no longer being able to continue to have the number of colonies with these types of losses that they're sustaining. And so this is bringing up a problem that beekeepers will eventually have fewer colonies in the US if things continue to go as they have been. Now, one of the big problems facing beekeepers is that there's no just one single problem causing all of these colony losses. It's a multitude of problems. It's things like parasites, it's poor nutrition, it's pesticides, it's pathogens, and all these things are acting synergistically to cause these really high levels of colony losses. But for my dissertation and for a lot of my PhD research, I'm really just going to focus in on parasites, and in particular, 2 parasitic mite species. So especially in the US, parasitic mites pose a current and future threat to honeybee colonies. Now, the current threat is a mite that we've been dealing with in the US since the 1980s. That is the Varroa destructor mite. But we're also seeing the potential to have a future problem with a mite species that's on the horizon and we're a little bit worried about, which is called Tropilaelaps mercedesae. And so these are really the 2 mites that are the basis of a lot of my PhD research and background. So just to give a quick overview of what my dissertation is going to entail, I have 5 different somewhat experimental chapters, not including my introduction and conclusion. So my chapters 2 and 3 are really going to focus on that Varroa mite control. And these are studies that I conducted in the US, looking at things that would reduce that Varroa infestation rate and the overall impact on some of the Amitraz resistance to a synthetic chemical that is used against Varroa, and seeing what the impacts of these different alternative treatments are. Now, both my chapter 2 and my chapter 3 are fairly similar. So in the interest of time, since I do have a large number of chapters, I'm really going to be focusing in on that chapter 3, which looks at a lot of these different alternative treatment strategies. Now, moving on, I'm going to transition a little bit in the middle of my talk and really cover Tropilaelaps. So I'll be going in, my chapter 4 is on the overall global range of Tropilaelaps. Chapter 5 is on how Tropilaelaps is dispersing. And then chapter 6 is a little bit on some Tropilaelaps management. So it's a little bit of a toss-up between these 2 mite species that are parasitic on honeybees. So starting out and getting into a little bit of a background on Varroa mites, this is coming from that same study that was showing those really high colony losses. This is showing that US beekeepers, it doesn't matter if they're backyard, sideliner, or commercial level, all of them are reporting that Varroa mites are a significant cause of colony losses within their operations. This isn't really a surprise to us. So a little bit of a background on Varroa biology. Varroa has two distinct phases in the colony, and they're capable of impacting honeybees in both of them. So starting out with their reproductive phase, a Varroa mite is going to enter a larval honeybee cell right before it's capped over for pupation. During that time where the honeybee is pupating, that Varroa mite is going to be constantly feeding on the hemolymph of that pupating bee. It's going to lay one male mite egg and then subsequent daughter mite eggs. Those daughters are going to go through an inbred breeding system. And then the goal for that mite is to reproduce and have daughter mites exit the cell with it. Now, when those mites exit the cell on those adult bees, they enter what's called the dispersal phase. So during the dispersal phase, the mites are usually on the adult bees. And this is kind of a heat map showing where the mites are predominantly being found on these adult bees. And you can see that mostly they're underneath the bottom segments of the abdomen of the honeybee, and they're constantly feeding on the fat bodies during this time. And so they're feeding on the hemolymph during the pupation. They're primarily feeding on fat bodies while they're out on the adult bees. And this is really impacting the overall honeybee because it's being fed on multiple times. Another thing that's really harmful to the honeybee is not only is this feeding being a detriment to honeybee health, but these mites are also capable of vectoring different viruses while they feed. And so you're seeing a detriment from the initial feeding, but also potentially from some of the viruses that these mites could be vectoring into these bees. And what's been found is that honeybees colonies, if they are not treated for Varroa mites, if there's no beekeeper intervention, usually these colonies end up dying in less than 2 years, and generally it's less time than that. If a colony goes untreated, it's probably not going to make it more than 12 months. So when we're talking about the different Varroa mite treatments and what's available, it's really treatment effectiveness is really dependent on the season and the colony conditions. Now, a lot of the treatments that we use for Varroa mites are going to be more contact-based treatments. So they're really going to need to come in contact with those Varroa mites. Now, this works really well if those mites are in the dispersal phase, if they're out feeding on those adult bees. Take, for an example, an oxalic acid dribble. You do that. You have mites out there in the dispersal phase that are feeding on the adult bees, you're going to get pretty good control and you're going to knock down most of those mites. Now, if you do that same oxalic acid dribble, but you do it when there's some levels of brood, if those mites are being found in those brood cells, that oxalic acid is not capable of penetrating that wax cell capping. Those mites are going to be protected and you're not going to get very good control. And you see, this is a bit of a conundrum with the honeybee industry as a whole, because we need high levels and high amounts of brood production, because we need to have those bees constantly churning out new workforces and new worker bees. But the problem is the more brood that you have in the colony, the more brood that's available for those Varroa mites to reproduce in, and the harder, if they're reproducing in that reproductive phase, it's going to be to actually kill those mites. So what a lot of beekeepers have turned to is extended release treatments. These are treatments that are valid in the colonies for an extended period of time, somewhere a lot of times between 42 to 56 days. And the goal of these extended treatments is that as those mites exit the reproductive phase and enter the dispersal phase, there's still contact treatments, but those mites are feeding on those adult bees. They're coming in contact with those different miticides, and then those mites are ended up dropping off and dying from coming in contact with those miticides over an extended period of time. Now, unfortunately, this continued reliance on these extended release, especially these synthetic chemicals, which are predominantly extended release, has led to some resistance, and especially resistance to Amitraz. Now, Amitraz in the last 20 years has been the most widely used chemical miticide for commercial beekeepers. And in the recent years, we started to see an increase in the overall levels of Amitraz resistance. And so I have a little graphic to explain how that happens. So here in this top left kind of corner, you see that you have a colony that has Varroa mites in it, and that one red mite constitutes an Amitraz resistant mite. Well, if you do an application of Amitraz as denoted by these square or these rectangles, then you're going to get pretty good control. You're going to knock down most of those mites, but that Amitraz resistant mite is going to be able to survive. So if you continue to apply Amitraz over and over again, eventually you're going to have later generations where you're going to have a lot more Amitraz resistant Varroa mites in that population. Then you do that same Amitraz based product, but you're not going to get very good control because those mites have developed a resistance, and so you're going to have to turn to some other form of treatment to control those Varroa mites. And so this is data published or unpublished, but data collected by Dr. Frank Rinkovich at the USDA Honeybee lab in Baton Rouge, and he's done Amitraz resistant tests all over the country. Now the x-axis isn't very important, that's just different beekeeper codes, but the y-axis is showing relative Amitraz resistance, and that black bar at 30%, that's essentially where we see a threshold where the Amitraz treatment is no longer effective. So even beekeepers that have that above that 30%, that's where we see when they do an Amitraz-based treatment, they're not getting Varroa infestation knocked down. They're not decreasing that Varroa infestation rate. And naturally, the higher Amitraz resistance that you have, the less effective that Amitraz-based treatment is going to be in your colony. Now, what's concerning is that Dr. Rinkovich has been doing this year over year, and he's seen that this is not a problem that's stagnant. This is a problem that's increasing. More beekeepers are reporting Amitraz resistance levels over this 30% threshold year over year. So this is becoming an increasing problem, especially in the commercial beekeeping industry. And so that's where a lot of my research comes in, is it really focuses on identifying alternative treatment strategies, things that are going to work in lieu of Amitraz. Because what we're trying to do right now is we're trying to avoid the pesticide treadmill. And what I mean by that is the pesticide treadmill is where you have a new chemical, and that new chemical is going to be fairly effective. It's going to be very good. But then there's going to be continuous treatments. And so that chemical is going to be used over and over again because it is effective. But eventually, the Varroa mites in this case are going to become resistant to that chemical. So what does a lot of times happen? Well, we apply more of that chemical. We apply it at a higher dosage because we're trying to still continually use that chemical. Well, that just makes the Varroa mites more resistant. So then we apply the chemical at greater frequency. We do more applications of the chemical. Eventually, Varroa mites become completely resistant, and you're no longer even able to use that chemical. Now, this has happened to actually 2 different chemicals for Varroa mites in the past. And what we're really trying to do is find something as an alternative to Amitraz before we make Amitraz completely useless, as we've done with a couple other chemistries in the past. And so that's where a lot of my research comes in my first Varroa mite chapter that I'll be focusing on today. And that's evaluating fall Varroa treatments and their impact on these Amitraz-resistant Varroa mite populations. So beekeepers often have brood late into the season in the southeastern US, and this creates a problem for a couple reasons. One, it means that the mites are continually able to reproduce in that brood. During that pupation cycle, those Varroa mites are able to continually reproduce and replenish that Varroa infestation rate, even if treatments are being put in the colony. The other big problem is that Varroa mites are being protected underneath those cell cappings when there's brood in the colony. So a lot of these contact treatments that are more rapid acting, they're not going to be effective because they're not going to be able to kill those mites underneath those cappings. And so I wanted to test alternative fall mite treatments to, one, evaluate the Varroa infestation on adult bees, but I also wanted to determine the impact on Amitraz-resistant Varroa mites, which we have some moderate levels of Amitraz resistance within our own operation at Auburn. And so to do this, I set up 5 different treatment groups. This was all done in 2 different bee yards around Auburn. I had 75 total colonies. I had 15 colonies per treatment group. All of the colonies, as you can see pictured, were housed in double deep brood chambers. And so for my first treatment group, which is not pictured, it was an untreated control. My next treatment group was Apivar, which has that active ingredient of Amitraz. I did 2 strips of Apivar per brood box for a total of 4 strips per colony. And I had those strips in the colony for a total of 42 days. I then had a Formic Pro, which has the active ingredient of Amitraz, formic acid, And I did 2 Formic Pro pads for a total of 14 days. Now, formic acid is a really interesting product because it's one of the only chemicals available as a miticide that's capable of penetrating those wax cell cappings. Now, with that becomes a little bit of a caveat. While it can penetrate those cell cappings and kill mites found within the brood, one of the problems is that it has a rather narrow temperature range window, and when used at higher temperatures, it can result in adult honeybee loss and sometimes queen death. So I had to wait until the end of September to be able to apply this when it came in the temperature range window in Auburn. My next treatment group was HopGuard, which has the active ingredient of hops beta acid. I did 2 strips per brood box for a total of 4 strips per colony for 14 days. I then went in after 14 days and reapplied another 4 strips per colony. So in total, they got 8 strips over 28 days. And then lastly, I had OA vapor, which is oxalic acid vapor, which has the active ingredient of oxalic acid. I did 2 grams of oxalic acid vapor per brood box for a total of 4 grams per colony. And I did a treatment regimen for this where I did a treatment every 7 days. So I did 3 total treatments every 7 days for a total of 3 treatments over 14 days. Now I assessed colonies on day 0, which was the end of September, day 63, which was the end of November, and then day 133 after an overwintering period in the beginning of February. And I assessed my colonies for capped brood and worker bee area, colony weight, and then I looked at the Varroa infestation on adult bees by performing alcohol washes. And one of the other really cool things is that I collected all of the Varroa mites that I found in these alcohol washes, and I sent them to Dr. Rinkovich in Baton Rouge, and he then had all of those mites genotyped for the Amitraz resistance allele frequency. So I'll be showing those results today. Now I want to point out that the colonies were removed from the study if they went queenless, the colony was weak, or if the colony had more than 45 mites in the alcohol wash on day 63. So why 45? Well, this generally means that the colony had roughly a 15% infestation rate of Varroa mites. This means that the colony at this rate is tending to trend downward and it's in possibility of crashing. And we were really worried about a high number of these colonies with high mite loads potentially crashing and then those colonies being robbed out and reinfesting the other colonies in our yard since they were housed in 1 of 2 locations. So any colonies that were over 45 mites on day 63, they were moved to a secondary location. And I was planning on assessing them on day 133, but all of those colonies ended up dying if I removed them because they did have extremely high Varroa mite infestation levels. Now, in the interest of time, I'm not really going to go in much to my colony strength metrics. I didn't see a whole lot of differences between any of the treatment groups on the different days. But I did see some differences in the Varroa mite infestation rate. So this is Varroa mites per 100 bees on the y-axis. And on the x-axis, you can see the difference of days. And so you can see on day 63, the Apivar, Formicpro, and the HopGuard were all significantly better than the untreated control group and the OA vapor group. But the other thing that I want to point out is that while they were significantly better than the control and the OA vapor, they didn't significantly reduce that Varroa infestation rate. So they were essentially just able to keep that mite infestation rate constant. While this is obviously better than the mite infestation rate spiking, it was a little disappointing that none of my treatment groups were capable of reducing that Varroa mite infestation rate when applied in the fall. Now, on day 133, we don't see any significant differences between any of the treatment groups. This is likely highly due to some survivorship bias. As I mentioned, all the colonies that had over 45 mites on day 63 were moved to a secondary location and they all ended up dying. Most of those colonies, as you'll see on the next slide, were control and oxalic acid vapor colonies. So you do have some likely survivorship bias impacting the results that I saw on day 133. So here's what the final colony status was for all the colonies in these different treatment groups. I'd like to point out that Apivar was the only treatment group that saw all the colonies being able to survive the entire length of the study. As I mentioned before, the control and the OA vapor were the 2 groups that had multiple colonies removed because they had really high Varroa mite infestation levels on day 63. Now, I want to point out Formicpro, particularly right here, because Formicpro performed equally as well as that Apivar Amitraz-based group for Varroa mite infestation rate. But you can see that in this group, 5 of my 15 colonies, 1/3 of those colonies lost their queen between day 0 and day 63. And this was a little bit frustrating to me because I purposefully waited for that formic acid application to come back into its temperature range window, but I still saw fairly high queen losses, higher than in any other group. And so when I'm giving this talk to different beekeepers around the Southeast, what I like to recommend to them is Formicpro can be an effective treatment used in your colonies, but I recommend using it in the spring when it's more easy to recoup a queen loss compared to the fall. Because if you lose your queen late into the fall, it's very difficult to requeen that colony, and you're likely going to have to combine it and count that essentially as a colony loss. Whereas in the spring, if you lose your queen, you have more readily available resources and queens that you could potentially requeen that colony. Now, finishing up with the HopGuard group, what was interesting about HopGuard is that it had essentially the same Varroa infestation rates as Apivar and Formic Pro, but it didn't really have any singular cause of why colonies were being removed. One colony had high Varroa, one colony had a queen failure, one colony was removed because it was weak. One colony, our technician accidentally got the queen in the alcohol wash, so it had to be kicked out. But things happen, you know. So it was really interesting to see that HopGuard performed fairly well in comparison to the Apivar and the Formic Pro treatments. The thing that I want to note with HopGuard, though, is that we were putting in 8 strips over 28 days. This is essentially the maximum dosage that you can have with HopGuard. And having some anecdotal conversations with beekeepers, they think HopGuard is too expensive. So we're only seeing these results with essentially the max dosage. And so beekeepers are a little bit wary of using HopGuard at a large scale because they're worried that of the overall cost if it's only capable of just maintaining their mite levels. So now as we transition, I'd like to move over to talking about the Amitraz resistance allele frequency results. So this is a little bit of a complex figure, but I'm going to more walk you through it. So the y-axis is showing you the relative genotype frequency of the different Amitraz resistance allele genotypes. And on the x-axis, you can see the different genotypes that were available. So this is showing the octopamine beta-2 receptor, and that Y215H is where the mutation occurs that allows these Varroa mites to have Amitraz resistance. So at the amino acid point 215 in the octopamine beta-2 receptor, susceptible mites are going to have an amino acid of tyrosine, which constitutes for the Y. But resistant mites are going to have a histidine. And what this mutation does is it prevents Amitraz and its subsequent breakdown products from binding to that octopamine receptor and killing that mite. And so you see, you have 3 different options. You have homozygous susceptible, which is the SS. You have heterozygous, which is that SR, and then you have the homozygous resistant, which is that RR. So when you look at the results from the Apivar, you see that an application of Apivar decreases from day 0 to day 63 the number of susceptible mites in the colony and increases the relative genotype frequency of the resistant mites in the colony. This is what we would tend to expect to see, because treating with an Amitraz-based product is going to naturally select against mites that have susceptibility. So it's going to kill any mites in the colony that are susceptible to Amitraz, but it's going to positively select for any mites that have Amitraz resistance. So mites that have that Amitraz resistance allele, they're able to survive that Amitraz application and survive throughout that application process. Now, what was interesting and kind of odd to see is that after that day 63, after that overwintering period, we see a drop down in the resistance and susceptible, and an increase in the susceptible resistance relative genotype frequency within these 2 groups. And so we don't really know what was the cause of that. We see it drop down. And one of the possible possibilities is that, well, maybe Amitraz resistance has a fitness cost, especially overwintering. And so what we'd expect to see with that Amitraz fitness cost is that maybe mites that have that Amitraz resistance allele genotype, they're not able to reproduce as well, or they don't survive over winter as easily. But in the words of Nate Bargatze as George Washington, if it were only so simple. Because when we look at all these other treatment groups, we didn't see that same appearance of that Amitraz resistance fitness cost. If we did have an actual fitness cost, what we'd expect to see is a subsequent decrease periodically over time of the RR in every single group. And especially if there was an overwintering fitness cost, we'd expect that RR and the 133, that blue bar on the far right, in every single one of these other treatment groups, we'd expect to be for that to be significantly lower in every group. But we don't see that. So it doesn't appear that there's any actual fitness cost to these resistance allele. But the other important thing to note is that we don't see any differences across any of the resistance genotype frequency. So what that means more positively is that these mites are not experiencing cross resistance. So mites that are resistant to an Amitraz-based product that have that Amitraz resistance allele, they're not also going to be resistant to formic acid-based products or hops beta acid-based products. They don't have that subsequent cross-resistant, which is positive to see. But I'd like to take us back to that apivar and what was really causing that decrease in resistance allele frequency from day 63 to day 133. Now, I've talked this over with multiple different honeybee researchers and Varroa mite biologists, and we don't really have a great understanding or an answer. There's a few potential causes that have been thrown around, like maybe this was just random genetic drift. Maybe there were not very many Varroa mites in the sample. These colonies were housed in the same 1 or 2 yards. So these mites could have been moving in between colonies and it just kind of all homogenized. And then we saw the loss of that effect. Maybe there's some sublethal effects of Amitraz where mites that have that Amitraz resistance allele, they're capable of surviving the initial treatment of Amitraz, but there's some sort of sublethal effect that maybe inhibits their reproduction or maybe causes a problem for them down the line. So you don't see that initial drop, but then eventually they die. There could be other potential causes, and this is something that I'm really interested in researching in the future. So overall, kind of the conclusion of this chapter is we really need to have a better understanding and have more research to understand the Amitraz-resistant Varroa mite dynamics. And what I mean by this is understanding what really caused that decrease from day 63 to day 133 in the Amitraz group. And then also, this is a question that I get a lot of times that I don't have a great answer to is, well, if we quit treating completely with any Amitraz-based products, how long until the Amitraz resistance genotype just leaves and then we don't have any Amitraz resistance within our Varroa mite populations? And that's a great question that we don't have concrete subsequent proof of being able to answer. There's been a few preliminary studies, but nothing that's been actually published on it. So now as I transition away from Varroa mites, I'd like to take you back to the very start of my PhD in January of 2023. And so I had originally come in and my goal for my PhD 2.5 years ago was, well, I'm going to help solve the Amitraz resistant Varroa mite problems. And obviously that's still a work in progress. But I did have the opportunity to go to a meeting where they were talking about a new mite. And so the beekeeping industry kind of got a little bit of a wandering eye to this new mite called Tropilaelaps. And so this was a new mite that was bursting onto the scene, getting a lot of notoriety and publicity. And so I wanted to kind of, and Jeff, being my advisor, had some previous experience working with Tropilaelaps in the past. And he offered me the opportunity to work with Tropilaelaps, and I really jumped at it. So the remainder of my dissertation will really be covering some of these preliminary knowledge gaps that we're trying to fill working with Tropilaelaps. So one of the first things I do when I'm talking about Tropilaelaps to any beekeeping group is I like to compare Tropilaelaps to Varroa. Most beekeepers have a pretty good understanding of Varroa, and hopefully after the first portion of my talk, you do as well. So there's some distinct similarities and differences between these two mite species. When we're talking about how they're the same, both these mites are parasitic mites on Apis mellifera, which is the western honeybee. They're both capable of reproducing within the brood, which makes them difficult to sometimes control because they're a lot of times protected with that wax cell capping while they're reproducing. And they're both capable of vectoring viruses. Now, once you have that similarities, you start to see some differences between these mites. For one, Varroa can feed on larvae, pupae, and adults within the colony. Tropilaelaps can feed on larvae and pupae, but they're incapable of feeding on adults. Varroa requires a short adult feeding phase post-mating before laying eggs. And what I mean by this is so after a daughter mite mates with her sibling in the cell and then reaches maturity, she goes out and will usually be in the dispersal phase for generally 5 to 7 days before she's sexually mature enough to go in, reinfest a new cell, and begin reproducing on her own. This 5 to 7 days offers a treatment window when maybe some of those extended release treatments are going to be able to come in contact with that mite. Now, Tropilaelaps, seeing as they don't feed on adult bees, they don't have the same post-mating feeding phase requirement. They're capable of exiting that cell with that adult bee, going in and within 48 hours, infesting a new cell and beginning reproducing on their own. So they're capable of reproducing at a faster overall rate than that of Varroa. Another thing that helps them reproduce a little bit faster is that Tropilaelaps has a 24-hour egg laying window compared to Varroa, which is 30. And then lastly, Varroa mites in similarly infested colonies between Varroa and Tropilaelaps, when they're both found co-infesting the same colonies, Varroa mites were found to be 50% non-reproductive, and Tropilaelaps mites were only found to be 30% non-reproductive. So what this is really meaning is that when these mites are both found in the same colony, Tropilaelaps mites are out-competing Varroa mites, and as long as they have access to brood, they're going to be able to become the predominant mite species within that colony. So here are these two mites side by side. You can see they're about the same in length. Tropilaelaps is about 1/3 the width of Varroa, and you can see Tropilaelaps has much longer legs, which allows it to move around on the frames much quicker. It does not move around via bee as much as it moves around by itself, moving around on those frames using those legs. So Tropilaelaps damages colonies in a fairly similar way to that of Varroa. They feed on the hemolymph of these pupating bees, capable of also vectoring these different viruses. And it was found that bees that emerge from a cell that was infested with Tropilaelaps, they have a reduced overall lifespan, they have a reduced weight at emergence, and they're more likely to have some form of deformity, whether that be from Varroa feeding or from some form of virus or from Tropilaelaps feeding, or by some form of virus that Tropilaelaps mite could have vectored into it. So my chapter 4 is really looking at the global range of Tropilaelaps, because as the popularity of Tropilaelaps research increases, there's a lot more push to figure out where this mite currently is. And so first, I think it's important to understand that Apis dorsata, or the giant honeybee, is believed to be the original host of Tropilaelaps. So it was believed that Tropilaelaps' native range is essentially about in the same area that the Apis dorsata native range is. But Tropilaelaps has interestingly become found and confirmed in areas outside the native range of that giant honeybee, Apis dorsata. And so that brings up the question of where is this mite currently at? And with the increase in interest, there's a lot of differing reports. There's different publications that say different things, and there's different unsubstantiated, unreally documented reports saying, beekeepers saying, hey, I have this mite in this country, but we have no real way of confirming that. So what I wanted to do is I wanted to provide a comprehensive list of countries and regions that had been confirmed with Tropilaelaps. And so to do that, I went through the peer-reviewed publications because I only wanted things that had gone through the peer-reviewed publication process. And I was looking for publications that had confirmed Tropilaelaps by either molecular confirmation using DNA or morphological confirmation by keying out that Tropilaelaps mite. And so here are the overall results of what we came up with, where this mite currently is. So this is the Tropilaelaps mercedesae distribution. And I also wanted to point out, this is showing which Apis species it has been found with, whether that be Apis dorsata, the giant honeybee, or Apis mellifera, the western honeybee. Now, there's a couple things that I really want to draw your attention to on this figure. I want to draw your attention to that top left portion showing Russia, Georgia, and Uzbekistan. These are all confirmations of Tropilaelaps that have been published in the past year and a half. And these are all outside the native range of Apis dorsata. So we are seeing that Tropilaelaps is expanding outside that native range and is able to subsist on Apis mellifera bees outside of that native range. Now, the other thing that I want to point out is look at all that gray that's in there. There's a lot of area that has no reports of Tropilaelaps. And so this is something that we've kind of come across, is that there's a lot of undocumented and need for people to go in and do sampling in these regions that don't have any documentation of Tropilaelaps to determine if this mite is currently located there, because it's going to be able to bring us a better understanding of, one, how they might be able to control it, and two, how that mite might be able to be spread. Thinking about how that mite might be able to be spread, that's a big question that I get, is how does that mite move around in between colonies? And that was a really big focus of my 5th chapter, which is trying to find Tropilaelaps mites on exiting adult honeybees. And so when we're thinking about Tropilaelaps dispersal, it's believed to disperse similar to that of Varroa, but actually in reality, not very much is known about how Tropilaelaps disperses. So when I say that it's believed to disperse similar to Varroa, that means it's believed that it's dispersing on the backs of adult bees or on adult bees, and the adult bee will leave the colony with the mite on it, and it might drift and come back into a new colony. And so then it's being able to be established there. More likely, though, what's happening is that you're having colonies with really high infestations of Varroa. Those colonies are then being robbed out by surrounding stronger colonies. Those Varroa mites are then hitchhiking back on those robber bees, and then those Varroa mites are able to begin reproducing. Now, it was believed Tropilaelaps was known to disperse in the same way, but we haven't had any documentation of Tropilaelaps mites being found on adult bees exiting the colony. And so that's really what I wanted to try to do. So I went over to Thailand to do some of this research, and I established funnel traps on some colonies to study Tropilaelaps dispersal on adult bees. So this is what the publication that really gave me the idea to do it. I always like to show the picture. This is what the traps are supposed to look like. This is what the funnel traps should look like. We were dealing with what we could get through customs. So this is a little bit the Great Value Walmart brand of the funnel trap. But you can see that here I have the trap set up, and it was moderately effective. We're just trying to collect some bees that are exiting the colony, because my thought was, well, if I can find a single Tropilaelaps mite exiting that colony on one of these adult bees, then it's possible that that mite could be dispersed through some form of drift or robbing behavior. So in July and August of 2023, I collected 7,250 bees. I washed them all, and I found 0 mites, which was a little bit discouraging. But what I really want to draw your attention to here is that the colonies that we were working with had an average Tropilaelaps cell infestation in the brood of 0.75%. That means less than 1 in every 100 cells that we opened up when we were looking in these colonies actually had a Tropilaelaps mite in it. So when you think about Tropilaelaps mites, well, they don't feed on adult bees. They don't really have any need or reason to be on the adult bees. And if they're at really low infestation rates in the colony, then why would they just not be reproducing and trying to get those populations up? So I went back in January of 2024 to Thailand, and I wanted to say and see, okay, if I put these on really, really high infestation colonies, then can I see this kind of behavior being documented? And lo and behold, mites were found on the exiting adult bees in January. And so when I show you this table, I washed about 1/3 of the number of bees, but I found 18 mites. And most of those were coming from a singular colony. The other thing that I want to draw your attention to, though, is that the average Tropilaelaps cell infestation was 25.5%. So that means essentially 1 in every 4 cells that we were opening, we were checking for brood, had a mite in it. These were highly infested colonies. And so that begs the question, okay, I don't see this behavior at really low infestations. I see it at really high infestations. Is there some kind of equilibrium where you start to see them exhibiting this behavior? Now, unfortunately, I didn't have the opportunity to do this research, but my colleague, Dr. Dan Urel, went over to Korea, and he did some of this research, and he's hoping to publish about that and seeing if there's a threshold where you start to see that mites are being found on adult bees exiting the colony. And so what this work really showed is that Tropilaelaps have multiple means of dispersal to different colonies. A lot of times we think about Tropilaelaps mites being dispersed through beekeeper-mediated dispersal. Of course, Auburn's not doing the beekeeper-mediated dispersal. We have highly intensive quarantine protocols after working with Tropilaelaps, as I'm well aware. But we're knowing that probably what we're seeing is that in a lot of these cases, well, colonies are being moved into these regions, these colonies are infested, and so beekeepers are doing a lot of the movement. But this work also showed that the mites are capable of dispersing on their own, potentially whether that be through drift or robbing behaviors. So lastly, I'd like to finish up with my chapter 6 and my final experimental chapter, really looking at managing Tropilaelaps through combined cultural and chemical controls. And so when we're thinking about how mites could move in between colonies, in between different regions, I always get the question, okay, what happens if the mite is introduced to the US? And so there's kind of a worry with Tropilaelaps introduction is that we don't have a current eradication strategy. So I like to take the scenario, worst case scenario, what would happen if a mite were to come in during the almond pollination in California when you have millions of colonies in the area? Well, those colonies are going to disperse themselves, and if the mites have been able to disperse, all of a sudden you have mites up in Washington, you have them in Florida, you have them up in the northern Great Plains, you have them on the East Coast, and pretty soon you have the mites all over the US. This is essentially somewhat what happened with Varroa mites, where there wasn't an early detection, and then within 5 years the mite was pretty much everywhere in the continental US. But what would happen if you're able to get rid of that first mite, that first incursion? Well, in theory, all the other mites would then disappear and you'd be able to eradicate it. But that's a little bit easier said than done. So what I was tasked with working with USDA APHIS was, can we determine a management plan that eliminates all the Tropilaelaps mites from the colony? So if we do find Tropilaelaps mites, we can do this some kind of management or treatment plan. We can treat all the colonies in that apiary and in some surrounding apiaries, and we're reasonably confident that we're able to kill all the mites in that colony. Well, first you need to have something that's actually going to kill the mites. So what we know is that brood breaks have been shown to be very effective at killing Tropilaelaps mites. Now, this shouldn't come as a shock to most of you because Tropilaelaps mites can't feed on adult bees. So if you remove the larval and pupae from the colony, you're removing that food source. Those mites have been shown to not be able to live more than 6 days without the removal of larvae and pupae from that colony. Now, brood breaks have been shown in multiple different studies to be effective against Tropilaelaps, but they don't give that 100% efficacy. And remember, if we're trying to get that eradication program, we need to eliminate all the mites from the colony. And so I wanted to see what happens if you take that backbone of a treatment plan with a brood break and add an additional chemical treatment on there. So I did this research over in Thailand, and I established 4 treatment groups where I tested for Tropilaelaps. I had a brood break only treatment group, a brood break plus formic acid, a brood break plus oxalic acid dribble, and then an untreated control. And so we purchased colonies from a local beekeeper in Thailand and assessed them for adult population, capped brood area, and then Tropilaelaps infestation. And I looked at Tropilaelaps infestation in 2 ways. One of them was uncapping 200 brood cells per colony, and the other one was looking at mite fall on sticky boards. In my brood break colonies, I caged the queen on day 0 and released her 24 days later. Why 24 days? Well, a drone honeybee or a male honeybee takes 24 days to develop. So in theory, if that queen had just laid an egg, a drone egg, then 24 days later, that drone would have been mature enough and it would have emerged. And so I shouldn't have any brood left in that colony. Then on day 24, I also applied my treatments. I did just 1 pad of Formic Pro for 10 days, and then I did a 40 milliliters of oxalic acid dribble. And then again, I assessed colonies for colony strength and Tropilaelaps infestation on day 60. And I always like to joke that no matter where you are in the world, whether it's the US or in Thailand, the undergrads always get to help you with the grunt work. So I'm not going to really show much of my colony strength metrics. There wasn't a whole lot of differences, but we did see significant differences in the percentage of infested worker brood cells. So this is pretty much the percentage of infested brood cells with Tropilaelaps. And what you can see is on day 0, I was really worried. We saw really low initial levels of Tropilaelaps brood infestation. I was calling Jeff and I was like, there's no mites here. I don't know what I'm going to do. But we saw that after 60 days, we see a huge jump. So the untreated control. The untreated control went from less than 1% cell infestation to about 15% cell infestation. And we were seeing some of these colonies in this untreated control were actually succumbing and crashing down and dying because of really high Tropilaelaps infestations. Now you can see in the brood break and the brood break plus oxalic acid, we got really good control, but we didn't get that 100% efficacy that we wanted because we did see a couple mites. Now in the brood break plus formic acid treatment group, you don't see a bar there. That's because we didn't find any mites in any of the brood cells from those colonies, which was really encouraging. That's the basis of our eradication plan. But unfortunately, when you look at the mite fall on sticky boards in 24 hours, you see the same trend. We're pretty low mite fall on day 1, 12, 25, a huge spike in the untreated control that just continues on to day 60. On day 50, again, no mites in that brood break plus formic acid treatment. But unfortunately, on day 60, we did find some mites in that brood break plus formic acid colony. So in some of those colonies. So we couldn't say that any of our treatments were 100% effective at getting rid of all the mites in the colony. So what these results really showed is that brood breaks are extremely effective. The brood break alone is capable of really reducing and keeping those mite levels extremely low for Tropilaelaps. And that additional chemical treatment will maybe have slightly marginalized or marginally increased the overall efficacy of those treatments. None of them were able to eliminate all the mites. Now, I want to point out that all of these colonies were housed in the same location. So as I just showed on my chapter 5, mites are capable of moving in between colonies or being found on adult bees exiting the colonies. We had some really high mite infestation colonies in this yard, so it was possible that some of our colonies in these different treatment groups were being reinfested in this yard. And so that brought up, does treating the entire yard give you 100% efficacy? So if you do this fruit break plus formic acid treatment, if you do blanket the entire yard with it, will that give you 100% efficacy? So I actually went over and did part of this study in Thailand this past fall, and I'm parsing through that data right now. So overall, just some high-level conclusions for my dissertation. I think it's important to know that honeybee losses remain high, and a lot of that is due to consistent Varroa mite pressures. Varroa mites are capable of vectoring multiple viruses, and they're really damaging to honeybee colonies as a whole. And it doesn't matter what level you are, they're impacting all these honeybees and beekeepers. It's also important to know that we have a real big need, especially in the southeastern US, for fall Varroa treatments. You can see that with my results, yes, I kept mites steady at Varroa mite or Varroa destructor infestation rate and Varroa mite infestation rate, but I wasn't capable of reducing it. I didn't reduce any of those infestation rates, which is a little bit difficult to see because the southeastern US is really hard to control those mites with the level of brood that you have later on into the year. We also know that we need a continued research on Amitraz resistant Varroa mite dynamics to understand how long you need to treat with alternative treatments to get rid of that Varroa or Amitraz resistance genotype, and also to understand what really happened between day 63 and day 133 in that apivar treatment group. Now transitioning to Tropilaelaps, we really have to need to fill in knowledge gaps regarding the Tropilaelaps range. We have a lot of gray area that we still need adequate Tropilaelaps monitoring in. And then overall to understand how these mites might be dispersing into these new regions. And then lastly, there's a need to continue researching Tropilaelaps treatment options to really understand if this mite is introduced, do we have a viable and potential eradication strategy or eradication plan that we could do to these colonies to help prevent that mite from establishing. So with that, these are two of my publications that I already have. I believe the top left one with the QR code is Tropilaelaps dispersal. The bottom right one is the management study that I did. I'd really like to thank all of the funding sponsors that supported me, the Alabama Agriculture Experiment Station, Alabama Department of Agriculture and Industries, Project APIS M, and I was a Project APIS M Costco scholar, the USDA APHIS, and USDA ARS, who supported my projects in some way. And with that, I have so many people really to thank. First, I'd like to start by thanking my committee, Dr. Jacobson, Dr. Graham, Dr. Bruckner, and Dr. Rinkovich. I really appreciated their advice, their patience, and their willingness to be flexible with me on my overall graduation date. I'd also like to thank Dr. Huber for serving as my university reader during this process. I'd like to thank the entire Auburn Bee Center, as we're a center now. And within that, I would really like to thank Dan, who's been a great collaborator, friend, and mentor to me. Jennifer, who started her PhD at the same time as me, and has been really fun to be able to hang out with. And then, of course, the lab manager of the Bee Center, Kristen, who is always the life of the party and keeps the center functioning. I'd also really like to thank the Melaponini and Apini Research Laboratory, and in particular, Ajam Baum, who was my colleague and who I worked for in the lab over in Thailand. She was very welcoming and helped make sure that all of my research functioned smoothly. And then lastly, I would really like to thank my advisor, Dr. Jeff Williams, who provided me with so many different opportunities that I'm extremely grateful and thankful for. Lastly, I'd like to thank just the friends and family who have been with me this entire time, these past 2.5 years. And with that, I will open it up to any questions, as you can see some of my fun photos from during my PhD time. Thank you.
Okay, let's make the people on Zoom feel welcome. Are you able to see their questions?
Yeah, I think so. I can.
There's a couple there right now, so maybe read them out as well as you digest them, just so that the audience understands.
Yeah, so one of the questions is, are there other IPM strategies to reduce these mite populations other than miticides? That is a great question. Yes, there are some different strategies. One of them would be called drone brood trapping. So drones, which are the male honeybees, they take 3 days longer to develop. And so Varroa mites have a preference for drone brood because they're able to reproduce more mites. And so there's a strategy to do drone brood trapping where you put frames that are exclusively made into drone brood into your colony. The queen will lay drone eggs, the mites will go in, those cells will be capped over, and then you can remove that frame, freeze it, and theoretically kill all those Varroa mites. Now, unfortunately, some of the problems with things like drone brood trapping is that it's very time and labor intensive, and it's difficult to do at a large scale. It's also not proven to be extremely effective, drone brood trapping on its own. It takes a lot of different time. Another really good IPM strategy is there is becoming more Varroa resistant hygienic stock to Varroa mites. And so these are bees that are able to detect the brood like they are able to detect brood pheromone and be able to see when that cell is being infested with a Varroa mite. So they're capable of removing that developing pupae, and that stops that Varroa mite reproduction cycle, and it prevents that Varroa mite from reproducing. So that is kind of on the new forefront with this kind of these hygienic behaviors and these just resistant bees. That is increasing in development, but it's really not being able to be used at a super high, large scale just because there's limited available genetic stock. The other things that you can do for IPM strategies is
maybe let's pause.
Okay.
So that came in during one of your Tropilaelaps chapters.
Oh, okay.
I'll pivot a little bit real quick for IPM for Tropilaelaps.
Yeah, so IPM for Tropilaelaps is a little bit easier to do because the mite can't survive on the adult bees. So you can just create a brood break in your colony, and while it might not be able to kill all of the mites in your colony and give you the eradication strategy, it's capable of reducing that. So inducing your own brood break instead of using a chemical is a potential option for you to use with Tropilaelaps. As far as hygienic behavior goes for Tropilaelaps, that's still a very big unknown. We don't really know if bees that are hygienically resistant to Varroa are also going to be hygienically resistant to Tropilaelaps. So the next question is, 100% efficacy is a necessary but highly ambitious goal. Are you worried at all about receiving a false positive, reporting 0 mites when there might be 1? What kinds of checks do you have in place to make sure you can be confident reporting 100% efficacy of any treatment? So that's a really good question, and something that we've been parsing through, and I've had some conversations with. The biggest issue is the only way to be able to more definitively say that we killed all the mites would be to freeze the colony and kill the colony, and then go through every single brood cell and check the bottom boards to see check every bee to see every single bee, the bottom board, go through all the debris and check all those cells. And so in lieu of killing the colonies, this is kind of more of a preliminary study to see, well, could we even do it at this smaller level scale to prove? And so we have been insufficient at doing that. I think that if we did find a strategy that we found at this smaller level scale, maybe we could then ramp up and start doing more colonies and do a more thorough inspection looking at that. But right now we're still kind of parsing through, can we even try to find a way to kill all the mites at the levels that we're looking at?
So let's take the next two on Zoom, and then we can transition to the.
Okay. Any thoughts on potentials for Varroa synergistic interactions, competition facilitation? Yeah, so this is an interesting question when thinking about how they are found in the same colonies. Right now, there is possible potential for different synergisms with those mites because they're both being found in the same colony. They're both impacting those colonies. What's more likely is that depending on your region, you might become and see that one of those mites becomes the predominant mite species being found in those colonies. So what we found during our research during my PhD is in Thailand, the colonies have brood year-round. So those Tropilaelaps mites are able to continually reproduce at a very high rate. and they've actually outcompeted Varroa. And so Tropilaelaps is the predominant mite species in Thailand. Now you take that to a more temperate area that has more intermittent brood periods and periods where they have a small brood break, like South Korea. Well, in Korea, you still have a large problem, and Varroa mites are the more predominant mite species in that area. So if you extrapolate that over to the US, I think it's going to be very regional dependent as to which mite you're going to have more of a problem with. And it's going to be also somewhat beekeeper dependent, as in, are you moving your colonies up north and then bringing them down south? Because then you're potentially allowing year-round production for Tropilaelaps. Whereas maybe if you do more overwintering storage, you reduce the overall levels of brood in your colonies, and then you're not going to maybe have as big of a problem with Tropilaelaps, and Varroa mites still might be your large issue. So competition, yes, they will compete for the same area, but they can coexist in the same colonies at the same time. And then last question that we'll deal with on Zoom. Many commercial beekeepers manage bees in both warmer climates and cooler climates, and your career will give you a chance to research mite control in both climates. Can you comment on how moving your research activities to North Dakota will change the opportunities you expect to have for research on Varroa control? Yeah, so this is a really interesting question. And so one of the things that was eye-opening to me moving down to Auburn is really the limitations that you have on the number of available products to use against Varroa. And so whereas up in Kansas and Nebraska, where I previously was, is you generally have more of a brood break, or you have a brood break period more earlier in the year. So what that allows you to do is it allows you to go in and treat for any of those mites in the dispersal phase. Now down here in Auburn, you sometimes will have brood year round. It's a study that we've been looking into. And so the number of different treatment options that you have is a lot more limited. And so thinking about moving up to North Dakota, I'd like to be able to, one, look at how they're overwintering bees in these sheds and look at to see if there's any opportunities of putting in treatments either in these different sheds or previous to sheds to be able to knock down those mites so they're ready to come out when they're going to do that commercial beekeeping pollination out in California. The other thing that I'd like to do more of is probably potentially a little bit more formic acid work. I know that formic acid can be very effective. And also we know that formic acid in the Southeast is you're a little bit more hamstrung as to when you can apply it. Whereas I know people in the more northern climates, especially one researcher in Pennsylvania, who absolutely loves formic acid and recommends that as one of her #1 treatment options. And so having that opportunity to have a few more things at my disposal will open up that toolkit a little bit more when working with some of these commercial beekeepers.
Yeah. Any questions from within the room here?
Yeah, Dr. Hill.
So, good presentation, Rogan. I enjoyed it. Bees are known to be hygienic, right? I mean, they're constantly, there's cleaning going on. So at Tropilaelaps, are there any hygienic behaviors in the colony that can safeguard against this initial, but going back to the question where, like, what happens if one just gets in? Are there any safeguards initially in bee behavior against that initial colonization or the initial introduction or incursion of Tropilaelaps?
Yeah, so the question is, bees are known to be naturally hygienic. Is there any way that the bees might naturally be able to have some hygienic behavior that's going to help mitigate that first incursion of the mite? I don't know entirely, but I would predict probably not. From my understanding and my work working with these bees, at least the bees with the hygienic behavior in Thailand. Of course, it's different, maybe depending on which region the mite were to be introduced, where it's introduced at. But we didn't really see problems in any sort of hygienic behavior with honeybees until those infestation rates started to get a little bit higher. Then we started to see some hygienic behavior like quote unquote bald brood, where they're opening up those cells and removing those pupae that they find infested. We are seeing that at rather high infestations, not at the level of a singular mite. Now, the other thing that I'll point out is that there is a study that's been done looking at bees grooming and the difference between 3 different honeybee species. Apis dorsata, which is the giant honeybee, Apis mellifera, which is the western honeybee, and Apis cerana, which is the Asian honeybee. And they found that by far and away, Apis mellifera, the western honeybee, is the worst at like grooming themselves and finding like the mites on them. Apis dorsata and Apis cerana were much better at grooming the mites off them, and they were much more likely to damage those mites during that grooming process. And so unfortunately, it's created a really naive host, as Apis mellifera seems to be the easiest one to infest. Yeah, Anthony.
It's like some like CRISPR, just like slap that in there. You can do that. Like identify like genes or like the drives that like have behavior, kind of modify their genomes. Like that.
Yeah, so the question's on CRISPR. That's really interesting. That's there is a lot of new work, especially with like the honeybee pangenome coming out and understanding the different genomics of honeybees. I think that's work that can be done in the future. We're also seeing more technological innovations, like things against Varroa mites, like RNAi being used as a treatment and a mode of action with double-stranded RNA. But we're not really, I don't know exactly if that would be possible at the current rate. I don't know of any research off the top of my head that's doing that, but that's a really good question. Yeah.
Excellent presentation. So this is a question from somebody who is not in the water totally. So you mentioned that you see it resistant to Amitraz, whatever the compound in Auburn, where your plants here.
Yep.
So where are they coming from? Are you actually applying this compound before you look for the resistance? How do you find it here if they are not exposed to the compound?
Yeah, so the question is really, we already have Amitraz resistance in Auburn before we even applied the compound. So how did we get that Amitraz resistance? That could have happened in a couple of different ways. For one, it's pretty common to have some at least low levels of Amitraz resistance in a lot of these different operations if you've used Amitraz really in the past. And if you use Amitraz, one time a year, you're going to probably keep some level of that if you've gotten it into it. The other thing is potentially you have some of these, depending on where you might be purchasing your colonies, you could be purchasing colonies that might have some Amitraz resistant Varroa mites already in them. And so then you have those colonies that you purchase, and now you just are propagating at some levels, small levels of Amitraz resistance. And as I said, if you treat with Amitraz, like you treat with an Apivar strip, then you're just going to be positively selecting to keep at least some level of Amitraz resistance genetics within those colonies. Yeah.
Any documentation on Tropilaelaps being found on other non-Apis social bees?
Yeah, so the question is, has Tropilaelaps been found on any non-social Apis bees? Or non-Apis bees, yeah, non-Apis social bees. I don't know of anything off the top of my head. I know that they've been found in Apis cerana, Apis dorsata, Apis laboriosa, which is another giant honeybee species, but non-Apis species, I don't know if there's been any documentation. With that being said, I don't know if there's really been that many people looking for it either. So they could be, and we just haven't seen it. It's not that they can't. Yeah, that's right.
Rogan, I have two questions. First question is, picture upper, two pictures from the right.
Yeah.
I need to know where you got, where I go get a pair of those pants.
In Thailand, the Thailand night market.
My other question is, I don't know how popular the treatment is for here. I think in Europe, there's something called a shook swarm treatment. You be familiar with that? It's basically where you shake out, shake off all of these off the frames and have them fly back to the spot where new frames are set up, all brooders are removed. And I was wondering, with Tropilaelaps seeming to be less present on the bee body, if that is a strategy that's attempted in places where Tropilaelaps is, or what do you think about the effectiveness, possible effectiveness of the strategy?
Yeah, so I don't know about the effectiveness of that strategy being to eradicate, but that would be an extremely effective strategy just in general for Tropilaelaps. And we've seen that beekeepers in Thailand do a variety of different things. So naturally, they do some forms of chemical treatments. They really like formic acid because, of course, it penetrates those cell cappings. It kills those mites in the brood. They have some other synthetic chemical treatments that they use, but they have some beekeepers that will just pull out brood frames and just burn them. And then it's somewhat similar to that shook swarm method where they're removing frames of brood, and they would rather take the hit on the strength of their colony and the hit on the potential honey production to remove those mites from their colony. And so the big thing that you see in a lot of these Thai colonies as well is that these colonies they're like much smaller colonies. And so they don't have near the amount of brood that they're being produced, which helps artificially keep mite levels down, because of course, the more brood, the more chance that those mites have to reproduce. And then the more mite infestation you're probably going to have. So Thai beekeepers have a few different strategies. One of them is they keep those colonies really small, but yes, they will not usually perform a shook swarm method, but they will do a kind of byproduct of that where they'll take those frames of brood out and essentially kind of like drone brood trapping, we'll just burn them instead.
Okay, I think we're gonna have to wrap up because we're a few minutes over time already. So let's congratulate Rogan on this episode.